Hi, everyone. Welcome to this week's GREG presentation. It's my honor today to introduce our speaker, Megan Barkdull. Megan got her Bachelor of Arts in Biology from New College of Florida in 2018. She then went on to do a PhD at Cornell University in the lab of Corrie Moreau. She began that in 2019, and she just defended this last semester, so congratulations, Megan, and is now becoming a PhD this semester and starting as an incoming assistant curator of entomology at the LACM, at the Natural History Museum of Los Angeles County, where she'll begin her job in January. So her research focuses on the evolution and development of complex traits in insects. We're all here to hear about complex traits in ants. And she's also interested, obviously, in natural history collection-based research. So I will let her share her slides and we'll get started. I just wanted to point out that this is a formal SSE event, and as an SSE event, we abide by the SSE Code of Conduct. So Robert is going to be posting a link to the Code of Conduct in the chat for those who would like to refer to it. Okay, thanks. Megan, take it away.
Great. Thank you so much for that introduction, Kelly. I really appreciate it. And thank you all for being here today. I am really excited to share some of my research with you today, exploring the evolution of complex traits in ants. Give me just 1 second. All right, sorry for that, shared offices. So getting into the talk, like probably everyone here today, I am really fundamentally fascinated by the tremendous diversity of life on Earth. And as an evolutionary biologist, what that means is at the end of the day, I'm really curious about all of the processes that have generated this phylogeny of life on Earth, the things that take us from a single-celled last universal common ancestor to the tremendous diversity of organisms that we see around us today. Now, of course, ecologists and evolutionary biologists study all kinds of different mechanisms that generate this biodiversity, from things like geographic isolation, which cuts off gene flow between populations and can lead them to diverge either adaptively or neutrally, to things like species interactions. So, for example, the specialization of parasites onto different hosts. The topic I've really spent a lot of time thinking about over the last 6 or so years has been the relationship between traits and patterns of diversification. And as you might have guessed, both from my talk title and from this slide, I've thought about this topic in the context of the evolution of one of my favorite groups of organisms, the ants. In part, that's because ants are tremendously diverse, with more than 14,000 species in 16 extant subfamilies, which have arisen since the last common ancestor of all ants evolved about 140 million years ago in the Cretaceous. And also because they have a ton of trait diversity. So here you can see we have variation in color, in mandible morphology. There's a lot of variation in sensory traits like eye size, and countless other traits pictured here. And so when I'm thinking about the relationship between traits and evolution in my research, I'm really guided by 2 big related questions. First, how do traits influence evolutionary processes? And I think about this question at macro scales, asking questions about how traits may be driving speciation and diversification, and also at more molecular scales, asking questions about how traits are impacting natural selection and genome evolution. Then on the flip side, my research explores how novel traits are generated, and I think about this by combining both evolutionary and developmental perspectives. Today, I'd like to share 3 stories that explore different aspects of these linked questions. First, we'll explore how worker polymorphisms may be impacting genome evolution across the ants. Next, I'll talk about my work using comparative genomics coupled with developmental transcriptomics to better understand the mechanisms that may generate worker polymorphisms in ants. And we'll wrap up by exploring some of the evolutionary mechanisms, origins of those mechanisms, All right, so let's dive into worker polymorphisms in ants. And my whole research program, and in some sense much of my fascination with ants as a group of insects, really stems from one remarkable fact of ant biology. So the 2 ants that you see on the screen right now are both members of the turtle ant genus, Cephalotes. And you can see that they have pretty distinct morphologies. The individual on the left is much larger, and she also has this kind of large disc-shaped head that smaller individual on the right lacks. And yet, despite these really obvious differences, these are actually both members of the same species. Not only are they members of the same species, they're both worker individuals from a single nest. And not only are they members of the same nest, but they have identical genomes. I think that's a really remarkable fact, right? Somehow development takes two genetically identical eggs, which hatch and grow as seemingly identical larvae. But via plastic development, they're able to pupate into these radically different morphologies and ultimately eclose as fully differentiated adults with distinct behaviors and roles in the nest. So that minor worker on the top is going to leave the nest to forage for food. She also performs most of the brood care tasks within the nest, while that larger soldier on the bottom pretty much exclusively performs nest defense. So she uses that large head to block nest entrances. And while I've used turtle ants as an example here, worker polymorphisms are fairly widespread in the ants. They're present in about 15% of ant genera and appear to have evolved at least 20 times. And they come in really diverse forms. So often they're simply size-based with kind of a bimodal distribution of worker body sizes in a single nest. We'll see this, for example, in carpenter ants. Sometimes these polymorphisms are based on allometry or the relative scaling relationships among body parts. So for example, in the Pheidole ants that you're looking at right now, that major worker in the center of the image is characterized by a really disproportionately large head relative to her body. More rarely, you'll have dimorphisms that are distinguished by a novel morphology, like that head disc of the cephalotes soldier that I talked about. And finally, we'll have continuous polymorphisms, where workers are spanning a really wide range of sizes, but with no real discrete binning. This is often the case in the leafcutter ants. Now, in all these cases, I really want to stress that these different morphs are the product of developmental plasticity. So there's no genetic determination of morph in any of these species. And we also know in many cases that these worker polymorphisms are providing really clear fitness benefits. So for example, in those polymorphic leafcutter ants, that continuous distribution of worker sizes has been shown to expand the array of dietary options that a single nest is able to take advantage of. And I also want to highlight that ants are far from the only group with striking polymorphisms that arise via developmental plasticity. So other examples include many butterflies, which can produce discrete wing patterns in different seasonal environments, reptiles that have temperature-dependent sex determination. And an example I really love are the spadefoot toads, which under crowded conditions develop into larger tadpoles with really robust jaws that actually allow them to engage in cannibalism, which is what you're seeing in this photo. Now, all of these really striking polymorphisms ultimately, to me, raise the question, how? How does one genome produce two or even more phenotypes? But again, because I like to think about the interplay of traits and evolution, it also makes me wonder what might be the consequences of this really striking plastic phenotype. And so while today I'll show you how I've addressed both of these questions in my research, I'd actually like to start by tackling the consequences of worker polymorphisms in ants. And this may seem like a little bit of a counterintuitive question, but we actually do have pretty clear evidence in a number of taxa that organismal traits can influence the genome. Two examples that I like to use here are generation time and parasitism, where organisms with shorter generation times accumulate a greater number of genomic changes over evolutionary timescales than do close but longer-lived relatives. And we know that organisms with parasitic lifestyles also tend to have elevated rates of genome evolution compared to their free-living relatives. However, of course, for most organismal traits, we really lack any clear understanding of whether they might impact genome evolution, and if so, how. And this is a really important question to understand because we also have evidence that these rates of genome evolution can either constrain or promote diversification. On the one hand, in classic adaptive radiations like African cichlids, clades with a greater speciation rate, here on the Y, also have increased rates of genome evolution on the x-axis. And then on the other extreme, some really fantastic recent work by Brownstein and colleagues demonstrates that in quote-unquote living fossils, including GAR, Rates of molecular evolution, shown on the y-axis, are extremely low compared to other lineages, potentially explaining both the extreme morphological stasis found in GAR lineages, and also the fact that species that are as much as 100 million years diverged are still able to successfully hybridize in this group. And so because we know that traits can impact genome evolution, and because genome evolution appears to influence diversification rates, I would argue that genome evolution is really a mechanism that we ought to be thinking about alongside other mechanisms like changing ecological opportunities or sexually selective pressures when we're thinking about how organismal traits can influence diversification rates. And so here I asked whether worker polymorphisms are influencing genome-wide patterns of molecular evolution. And I have two predictions. First, that polymorphisms will be associated with an overall decrease in the strength of natural selection. And second, that worker polymorphisms should reduce the extent of positive selection across the genome. Now, this first prediction, that polymorphisms are associated with relaxed selection, really stems from the fact that these alternative worker morphs are the product of plastic gene expression during development. Right? We know that in order to produce these divergent adult phenotypes from identical genomes, genes must be plastically turned on and off between morphs during their development. And in other systems, this kind of plastic gene expression is associated with relaxation of selection. And there are kind of two mechanisms that might explain this pattern. First, there's some evidence that a pre-existing history of relaxed selection actually enables genes to become plastically expressed, to become co-opted into these plastic phenotypes. And second, when genes are being expressed in only a subset of individuals, their effective population size is going to be reduced, which should strengthen the effects of genetic drift. And so together, these two mechanisms should manifest as relaxed selection at the level of the genome. My other prediction is that worker polymorphisms will decrease the degree of positive selection that we see in the genome. Why? Well, you can imagine that when you have these two morphs, which are performing really different functions in the nest, then selection might act to push traits to different optimal values in each morph. So as sort of a toy example, in these turtle ants, the minor worker on the left needs to leave the nest to forage, whereas the soldier is really staying in the dark interior of the nest performing defense. And so you could imagine that, for example, the optimal sensitivity of the eye to light might differ between these morphs. And so we might expect to see positive selection acting on the genes linked to this trait. But that tuning is really predicated on traits in each morph being independent of one another. If instead, traits are the product of the same genes in each morph, then selection won't be able to act on one morph without having some potentially detrimental impact on the phenotype of the other morph, resulting in what we often call caste-antagonistic pleiotropy. And in fact, we know from differential gene expression studies in ants and in other eusocial Hymenoptera that very few, if any, genes tend to be expressed in just a single morph. And so it's quite likely that many genes have the potential to be experiencing this kind of caste-antagonistic pleiotropy. And so here I'm really expecting that these polymorphisms impose a constraint to positive selection and should reduce its prevalence genome-wide. So to test these predictions, I used a large data set of 22 publicly available ant genomes, which you can see comprise both monomorphic and polymorphic species and represent about 140 million years of evolutionary divergence. Genomes in hand, I used a tool called OrthoFinder, which allows me to identify groups of orthologous genes across those genomes, and then used two methods from the Hi5 family of methods to explore gene evolution in association with polymorphism. I used a tool called BustedPH, which lets me test for positive selection linked to my trait of interest, and a tool called Relax, which lets me assess shifts in the intensity of selection associated with a trait of interest. So what did I find? Testing this first prediction that polymorphisms are going to be associated with an overall relaxation of selection, I'll show you these genome-wide results as a bar plot, where we're going to have selective regime on the x-axis and then count of genes on the y-axis. So most genes are showing no shift in selective intensity. And it turns out that roughly equal numbers of genes evolve under relaxed selection and intensified selection that's linked to these worker polymorphisms. Indeed, when we test for a difference in proportions here, there's no evidence that these proportions differ from equality. And so my first prediction does not appear to be borne out by the data. What about positive selection? Here, I'm expecting that polymorphisms lead to decreased rates of positive selection because genes may be experiencing cast-antagonistic pleiotropy. Again, looking at these genome-wide results of the bar plot, roughly 2,000 genes show no evidence for positive selection anywhere on the phylogeny. Then we have about 5,000 genes that are under really pervasive positive selection, but not specifically linked to the polymorphism. Getting into the genes we're interested in, a few hundred genes are significantly linked to worker polymorphisms, and it turns out that a much larger group are actually under positive selection linked to monomorphism, with these proportions significantly departing from equality. We see the same pattern being recovered for genes that are non-significantly associated with polymorphism and monomorphism as well. And so this second prediction does seem to be supported by the data, where I'm finding that these polymorphic species have significantly fewer genes under positive selection. And so while I didn't find support for polymorphisms being linked to relaxation of selection genome-wide, I do find that they seem to be associated with decreased rates of positive selection. And I think based on the results from this project, the key takeaway is that by creating these cast-antagonistic pleiotropies and potentially limiting the efficacy of natural selection, worker polymorphisms in ants may impose a genomic constraint to selection. Okay, so now having considered how these polymorphisms are impacting genome evolution across the ants, let's turn to the flip side and start to explore some of the processes that actually give rise to these worker polymorphisms. Returning to this original question of how. How does one genome produce two or more phenotypes? And I'm really interested in understanding this question both from a proximate perspective, thinking about developmental mechanisms, and from a more ultimate perspective, where we're thinking about evolutionary processes. Now, to address each of these perspectives, I've tackled this question using a combination of developmental transcriptomic approaches and comparative genomics, with the idea being that by combining these different approaches and these different lines of evidence, we can begin to identify some of the underlying mechanisms that actually enable this complex trait. Now, of course, in order to answer these questions, in order to do the transcriptomics, I needed some live turtle ants. And so I went down to the Florida Keys. I got to spend a lot of time in these really beautiful mangrove environments, getting distracted by the other cool creatures around me, like these Medusa jellies. And I should really thank the SSE, the Rosemary Grant Award, funded a lot of this field work. So thank you so much. But I also got to spend a lot of time snapping open mangrove twigs, hoping to see turtle ants looking back at me. So that's kind of what it looks like in the field. We're snapping mangrove twigs, and the colonies are inside those twigs. So then with turtle ants of different developmental stages back in the lab, I extracted and sequenced RNA from pupal and adult minor workers and soldiers. Now, of course, in an ideal world, I would have included developing larvae as well. But since we don't yet know how to differentiate between the two morphs at larval stages, that hasn't been possible yet. These transcriptomic data then allow me to identify genes that are being differentially expressed within a single morph during the course of its development, and also genes that are differentially expressed between these morphs during a single life stage, resulting in a total of 4 contrasts across which I can explore differential gene expression. And while there are a lot of really interesting results across all 4 of these comparisons, for the sake of time today, I'm just going to focus on patterns of differential gene expression between pupal workers and pupal soldiers. Now we can look at these results at a really broad scale in the form of a volcano plot, where we're going to have the fold difference in expression between workers and soldiers on the x-axis. So if a gene has a negative fold change, that's indicating that it's more highly expressed in the pupal workers, and a positive fold change would indicate higher expression in the soldiers. The y-axis then indicates the significance of the differential expression, and our individual points are also going to be colored by a p-value cutoff. So we have roughly 1,100 genes that are upregulated in worker pupae, and about 650 that are upregulated in the soldiers. Now, how do we make sense of these thousands of differentially expressed genes? Well, one option is to explore which well-characterized molecular pathways are overrepresented among the differentially expressed genes. So I'll present these results as a ridgeline plot, where each row in the plot is going to correspond to a significantly enriched pathway. The x-axis will again represent the differential expression of each gene in that pathway, same kind of scale as before. And the y-axis for each row represents the number of genes in the pathway with that degree of fold change. And then each plot is also going to be colored by the significance of the pathway's enrichment. So if we look at these results, what you can see is that we have 13 pathways that are enriched among my differentially expressed genes. Now, several of these pathways are related to brain or synaptic processes, and this suggests that there are probably neurological differences between the two morphs, which makes a ton of sense. They have really, really different behaviors as adults. But I'm actually most excited about this pathway in the middle, hipposignaling. And that's because hipposignaling is thought to help integrate between whole body signals of nutritional condition and imaginal disc morphogen signaling networks to help create a number of different insect polymorphisms. So for example, in these two beetle species, which have nutritionally sensitive head horns on the left and nutritionally sensitive mandible polymorphisms on the right, knockdown of components of the hipposignaling pathway interferes with the normal development of that polymorphic phenotype. And while we know that both nutritional signaling via things like insulin-like peptides and other pathways and the activity of imaginal discs individually are relevant to social insect caste determination, to my knowledge, this is the first time that hipposignaling has really been implicated as sort of an integrating mechanism in this process in ants. And so it may be the case that, as in these beetles, hipposignaling encephalodes acts to transduce the environmental or the nutritional inputs that are mediating morph differentiation. Now, of course, since we want to understand the relationship between those proximate developmental mechanisms that we just explored and more ultimate evolutionary mechanisms acting in this genus, I've also taken a comparative genomic approach to understanding this polymorphism. So we can use comparative genomics, of course, if we have a group with nice variation in the presence and absence of our trait of interest. So presence and absence of this worker polymorphism. And it turns out that cephalotes ants are a really great group in which to apply this kind of an approach. You can see, especially looking in kind of this part of the tree, that we have species both with and without that soldier morph, which I've actually found has probably evolved a number of times independently from a monomorphic ancestral state. And so for this project, what I've done is gone into the tree and select sets of paired monomorphic and dimorphic species. So we have these three phylogenetically matched species pairs. And here I'm hypothesizing that if this phenotype evolves via changes to protein coding sequences, then across the species with the polymorphism, we should find evidence for repeated natural selection acting on genes that are relevant to the phenotype. Alternatively, we may find no real evidence for selection acting on specific protein coding genes. And this could indicate that what's important here are changes to gene regulatory sequences when this polyphenism is evolving. And so to test these hypotheses, I first went in and sequenced the genomes for these cephalotes species, and then again tested all of their genes for two things: evidence of positive selection linked to this polymorphism, and changes in the intensity of selection in the polymorphic species relative to the monomorphic species. Now again, I'll show you these results as bar plots, where we're going to have selective regime on the x-axis and count of genes on the y-axis. And in particular, I'd like you to pay attention to these 4 bars, indicating genes with positive selection only in the dimorphic species, genes with positive selection only in the monomorphic species, and then genes with either relaxed or intensified selection in the dimorphic cephalotes relative to the monomorphic cephalotes. So taking a look at these results, you can see that we do have hundreds to thousands of genes that appear to be evolving in concert with this polyphenism. Now, to really better understand the relationship between the proximate developmental mechanisms that are producing plasticity and those ultimate evolutionary mechanisms that I'm picking up on with the comparative genomic approach, I asked whether these differentially expressed genes are evolving under selection more frequently than you would expect purely by chance. So the idea here is that we have these 2 sets of differentially expressed genes, those with minor biased patterns of expression, and then that smaller set with soldier biased expression. And then we can overlap them with the sets of genes evolving under intensified, relaxed, or positive selection across the 3 dimorphic cephalotes in my comparative analysis. Now, because we have 6 intersections that we're interested in, and because the bubbles are scaled to the number of genes in each set, these data are actually not very well represented by a Venn diagram like this. And so instead, I'll show you my results as an upset plot, which is going to have rows that correspond to those 5 sets of genes. We have a small bar plot on the left that's displaying how many genes in total are in each set. And then the intersections across those sets are going to be represented with a matrix with colored dots connected by lines. So for instance, this first column is going to show you the genes that are evolving under positive selection and which are upregulated in the worker pupae relative to the soldier pupae. So here, taking a look at the results for those overlaps, we have another bar plot displaying the number of genes in each of those intersections. And you can see that generally there are fairly few genes that are both differentially expressed and under selection, between 158 and 18, depending on the intersection. And when I test each of these overlaps to see if they contain more genes than you would expect purely by chance, only the overlap of minor biased expression and relaxed selection turns out to be statistically significant based on a hypergeometric test. And so in general, no, differentially expressed genes don't appear to be more likely to be under natural selection across this genus. And I think that what this is probably telling us is that rather than acting on the genes that are differentially expressed during the development of this polymorphism, selection may instead be acting on gene regulatory elements, which are the non-coding regions of the genome that help to recruit transcription factors and then mediate patterns of gene expression. And so based on that idea, as a final step here, I wanted to see if we can find any patterns in regulatory element evolution in these genomes. Now, this is harder than studying the evolution of protein coding genes, both because it's harder to identify these regions of the genome and also because we don't really have great models for how we should expect them to be evolving. But one thing that we can do is take the genomes from each species, and I'm showing you their phylogenetic relationships on the left. What we know is that these genomes are full of genes, of course, represented by these green bars, but also lots of non-coding DNA, which is represented in gray. And what I can do is align all 6 of my genomes and then identify non-coding regions that are highly conserved across the 3 dimorphic species, but which evolve at an accelerated rate in the monomorphic species. Now, because these regions don't code for a protein and yet are still really highly conserved across these long evolutionary time spans, remembering, of course, that these species last shared a common ancestor about 50 million years ago, we take that as evidence that these regions have some kind of a regulatory function and that is what explains their sequence conservation. We then interpret that accelerated rate of evolution as evidence of relaxed selection in the monomorphic species. I can then do the inverse, identifying conserved elements across the monomorphic species that have an accelerated rate of evolution in the dimorphic species, to find those regulatory elements that may be under relaxed selection in these species. Now, what I do is I perform these analyses across the genome, and this lets us get a sense of the distribution of relaxed selection on regulatory elements between these 2 groups of species. So again, I'll show you these results with a bar plot. We're going to have our 2 groups of species on the x-axis and then the count of those regulatory elements on the y-axis. Now, this is the count of total conserved regulatory elements found in those 2 groups of species. You can see that there are thousands of these elements. And then I'll overlay the elements that are evolving under relaxed selection. And what you can see here is that we have both a higher absolute number and a greater percent of these elements that appear to evolve under relaxed selection in the monomorphic species. Now, another way to think about this is that these are probably regulatory elements that are experiencing purifying selection in the dimorphic cephalotes species that may be relevant to the production of this polymorphic phenotype. And so this is really consistent with this model that selection, particularly purifying selection, is acting on regulatory elements more so than it is on particular genes. And so based on the results from this project, the key takeaways are that hipposignaling may play an important role in the development of ant worker polymorphisms, and that during the evolution of this polymorphism, selection is probably acting primarily on regulatory elements rather than protein coding genes. Okay, so now that we know something about the genes and the mechanisms that may be involved in generating these cephalotes polymorphisms, I'd like to wrap up by zooming out and thinking about some of the evolutionary origins of these mechanisms. So if we return to the tree of life that we started the talk with, and we start to think about our now best friends, the turtle ants, where did the genes that they're using to generate these polymorphisms come from? Are they genes that are found only in the turtle ants? Maybe they're genes that are found across all of the insects, but nowhere else. They could be genes that originated with the earliest of the protostomes. Or maybe they're genes that are found in all animals, or ancient genes that are present across the entire tree of life. And really, when we ask questions like this, fundamentally, we're asking, where does evolutionary novelty come from? Which is a really fundamental question in evolutionary developmental biology. Now, there's a lot of both theory and experimental evidence from Evo-Devo that can kind of guide our expectations here. So for example, if we're thinking about the evolution of butterflies, and we find a clade that has some kind of a novel trait, so in this case, eye spot patterns on their wings, we can ask how they're generating this novel trait. Did a new gene arise at the origin of this clade that actually allows them to develop eye spots? Or are they reusing some gene that's found in other butterflies, but co-opting or redeploying it to create this novel trait? And in this particular case, there's really pretty compelling evidence that eye spots develop by reusing or co-opting existing developmental genes, like the gene antennapedia, which is otherwise used to specify antenna development. And this is really a general trend where we very often see old genes being repurposed to generate novel traits. So we can see this at really broad phylogenetic scales, like the ubiquity of Hox genes being involved in regulating body plan development and diversity across the bilaterians. And so based on the finding that so-called toolkit genes, like these Hox genes, are repeatedly reused to generate things like novel body plans in animals, there's also been a lot of work trying to find kind of a similar toolkit for the genes that may be involved in novel social traits in social insects. Because it's really clear that sociality has evolved many independent times in hymenopterans, in ants, in honeybees, and in tons of other bee and wasp taxa. And so the social toolkit hypothesis is positing that across these different independent origins, there is repeated reuse of the same conserved set of toolkit genes when we're producing social phenotypes. Now, to test this hypothesis, folks generally take gene expression data from several social species and then test to see if there are very many overlapping genes across different taxa. There very often aren't. There tend not to be a great deal of overlap across these data sets. And then, instead of making arguments about shared genes, researchers will often point to kind of shared pathways and processes across those sets of genes. And they'll generally call that kind of the loose toolkit hypothesis. But of course, an alternative hypothesis is that each of these independent origins of sociality are underlaid by distinct or even novel genes. And that's certainly possible. There are some really compelling examples of novel traits being generated by truly novel genes. For instance, research demonstrating that the stinging cells of cnidarians are generated by the activity of a novel cnidarian-specific transcription factor. And so because the evidence for or against a social toolkit remains really inconclusive, here I'm asking, do cephalotes worker polymorphisms develop using primarily ancient or primarily novel sets of genes? Now, to answer this question, I used an approach called phylostratigraphy, where we can essentially classify genes on an ancient to young spectrum based on how deeply in the tree of life we can identify their homologs. For instance, if we can find homologs for a gene across all the ant genomes that we can look at, but nowhere else, we would assign that gene to the relatively young phylostratum Firmicidae. If a gene has homologs in a bunch of diverse insect genomes, it would be assigned to the somewhat older Hexapoda phylostratum. And if we can find homologs really broadly in animal and fungus genomes, then that gene would be in the more ancient Epistheconta phylostratum. So I first performed this classification process for all of the Cephalotes varians genes, which ends up looking like this. You can see that we have a significant number of these quite old genes on the far left, but also many young genes that appear to be Cephalotes specific. I'm now going to go ahead and log transform the y-axis of this plot. This will make the rest of the results a little bit easier to see. Now what we want to know is, where do the genes that are involved in worker morph development fall on this spectrum? And so here they are plotted in these darker colors. And what's important also to note is that the darker bars are colored based on whether the differentially expressed genes are overrepresented at a particular phylostratum in the green or underrepresented in the blue. And the overall pattern that you're probably noticing is that there's more green on the left of the plot and more blue to the right of the plot. In other words, that we have overrepresentation of these ancient genes and relative underrepresentation of these younger genes, indicating that these Cephalotes worker polymorphisms may be reusing a lot of ancient genes in their development. And while, of course, this alone does not prove that there is a genetic toolkit for worker polymorphisms, that would require more gene expression data from more taxa, from more independent origins of this trait. It is at least consistent with the predictions of the social toolkit hypothesis. All right, and so across these three stories, what have I kind of shown you today? First, we've seen that large-scale comparative genomics linking worker polymorphisms to genome evolution demonstrates that polymorphisms may act as a constraint to genome evolution. They appear to reduce the extent of positive selection across the genome. In Cephalotes turtle ants, we've seen that novel pathways like hipposignaling likely underlie worker morph differentiation. And finally, I've shown you that those morph-related genes are really disproportionately ancient, and that this appears to be consistent with some of the expectations of the social toolkit hypothesis. With that, I'll wrap up a little early. I'd like to thank my lab, my collaborators, the folks at the Cornell Insect Collection, and of course, all the funding sources that made this work possible, in particular, the Rosemary Grant Advanced Award from SSE. And I'll end just by sharing that I am starting as an assistant curator of entomology at the LA County Museum this January. So if folks are interested in working together or in putting together postdoc proposals, please do reach out. And with that, I would love to take any questions.
Thank you, Megan. That was amazing. So fun to see everything come together. We have plenty of time for questions. If anybody wants to ask one, you can either put something in the chat or just unmute yourself and speak up. Mary Jane?
Yeah, thank you. It was an amazing talk, and it had a lot that I'm not used to thinking about. And so this question, first I have to find out if I properly understood something. When you talk about the toolkit, are you really referring to the toolkit of regulatory genes? Is that correct?
Oh, that's a great question. I think when I'm referring to the toolkit, I'm kind of agnostic to what kinds of genes might be in that toolkit, but I certainly think it's quite likely that many or most of them might be regulatory genes. Mm-hmm.
Yeah. So one of the things that I'm especially interested in, and this doesn't mean, my ignorance of the toolkit questions doesn't reflect any judgment about the importance of it. What I'm used to thinking about is the origin of phenotypes. Mm-hmm. So I think I can conclude from what you said that what you're saying doesn't inform us really about where the distinctive phenotypes come from. It tells us how it was possible to get them out of the same genome. Yeah, okay, so I'm reassured by that. Because I was thinking at the beginning of your talk, there are really polymorphisms and polyphenisms in almost all organisms. If you look at immatures versus mature insects or humans or anything. So I wonder if in the future of your work, whether you will look at, I mean, it would involve looking harder, I guess, at the origin, at the patterns of expression of different proteins, really, protein-producing genes. So I wonder where you see the future of your research going. The other thing I wanted to just say is that it's wonderful that you're working with the collection as a collection-based person, because that's so important now, and not everybody pays enough attention to that job. So thanks for that really interesting talk and for what you're doing.
Thank you so much. Yeah, I mean, truly, this work would not have been possible without collections. So for example, those genomes that I sequenced in kind of the second chunk of the talk, that was during COVID. I wouldn't have been able to travel to collect those species myself. And yeah, it was only because they were represented in collections that I was able to do that project. So yeah, happy to kind of highlight that part of my research. And then your question about kind of where do I see this going next? I think that that's a really, really great question. I would really like to get gene expression data from other kind of independent origins that span more evolutionary time, independent origins of this worker polymorphism in ants. We could compare what's going on in the cephalotes to what's going on in Pheidole, and ask, do we see that the same genes are differentially expressed, or are they using totally different genes to build these different worker morphs? Yeah.
That'd be great, because basically what you outlined in the introduction of the different sort of situations in ants going from monomorphism within the morphologically monomorphic ones, there's behavioral diversity. I mean, so that you really outlined a huge project for the future.
Yes, yeah, lots and lots to do. And then another thing that I would have loved, and I wish I had gotten the samples during my dissertation research, but it just didn't work out, is to have also included developing queens in these data sets and be able to ask the question, the cephalotes soldier looks a lot like the queen. They both have that head disc. Is the soldier more similar to the queen, more similar to the worker? Kind of what's going on there is also something I'd really like to have the opportunity to explore further.
Yeah, that'd be great.
Yeah. Thank you so much for that question. Anyone else?
I have a quick question.
Yeah, Kelly.
I love these phylostratigraphy studies. I think that's kind of interesting. I mean, we're so curious to know, how far back are we talking when we're talking about how phenotypes get shaped over evolutionary history. I'm wondering if there's any way to parse down those overexpressed or underexpressed genes a little bit more. I mean, could we look at what genes those are and what pathways they're in, or do we not do that for a reason?
That's a really great question. Yeah, that's something that I'm still working on that project, and I am kind of trying to think about how do we draw additional meaning from those bar plots. I think certainly we can look at specific pathways that are enriched among those genes, even, just what are the genes themselves. Something I'd like to spend a little bit more time doing. You guys obviously saw that huge spike in cephalotes specific genes. And although we do often find that, newly sequenced and assembled ant genomes have a lot of species specific genes in them, I also think that a fair amount of that is probably because of assembly issues where genes are getting split across contigs. And so they look like they're cephalotes specific, but they're not. So I would like to kind of dig into that more. Yeah.
Any other questions for Megan?
I have a question.
Hey, Laura.
Hey, Megan. An excellent talk as always. Thank you for that. I'm asking this kind of selfishly, I guess, because I also am trying to figure out similar things. I was curious of when you were investigating the regulatory elements, and you concluded that the areas where there's a lot of change, you think it's relaxed selection. Is there any I guess I'd like to understand your reasoning. Not that I think it's wrong. I think you're probably right. of why
It's a
you
hard
think
question to
you're
tackle, though.
It's so hard, and so which is why I wanted to kind of pick your brain about it, because I guess because it could feasibly be positive selection, right?
Yeah.
Is this just because relaxed selection is so much more likely? Or maybe I can stop answering for you and you just answer.
No, that's pretty much it. Yeah. So essentially what I did is I identified these conserved regions across the 3 species that all share a particular trait value. Then I look at those regions in the other 3 species. And what I did to kind of get this accelerated rate is I compared the rate of evolution of those elements to the rate of evolution of fourfold degenerate sites in protein coding genes in those genomes. And so that's kind of like the benchmark. And so, yeah, you're exactly right. It could definitely be positive selection as well. And we're just kind of using relaxed selection as that null assumption of that's the most likely thing to be going on, especially across all of those elements. But yeah, in any individual case, it could totally be positive selection or like a combination, on different parts of the regulatory element itself. Yeah.
Of course.
Yeah.
Yeah, it's so tricky.
So tricky. If you're interested in kind of doing some of this stuff yourself, there's a really cool paper on killifish, actually, that's kind of where I got some of this method from. I would definitely recommend checking it out.
Yeah, I definitely would like to take a look.
Yeah. Awesome. Any other questions?
We run into this problem. I'm sorry, Robert.
You go ahead.
I was just going to say, we run into this problem also, Megan, of not being able to know what the phenotype is at really, really early stages of development. And you so want to know when those genes start deviating in their expression, right? Over developmental. And we've been toying around with thinking about doing sort of single cell stuff from really, really in our case, it's just really early juvenile species of lizards, where it's life stages of lizards. But yeah, that would be a fun challenge.
There's an incredible paper that I like. Their methods are so cool. I think from 2022, where folks were able to do kind of low coverage transcriptome sequencing for something like 2,000 developing eggs and larvae all throughout the developmental trajectory. And just with that scale of data, they're kind of able to reconstruct and trace back to before we can see morphological differences. Yeah, that would be the dream for sure. Super cool.
Yeah, I just want to say I like the lab logo.
Thanks.
No problem. But also, I was just wondering, can the regulatory regions be looked at between species? Can you make a comparison between regulatory regions that are the same across species? And then can you say if those regulatory regions developed, where they developed on the tree of life?
Oh, that's a super interesting question. So kind of doing the phylostratigraphy, but for the regulatory elements as opposed to the genes. Yeah, I mean, I don't see any reason that you couldn't do that. The thing that would be challenging and maybe kind of computationally expensive would be that because we can't necessarily just use, open reading frames and sequence similarity to identify homologous regions when we're working with these regulatory regions. I think what you'd probably have to do is generate like multi-species whole genome alignments. And that's what I did with my 6 Cephalotes species to find those regions. And so there's no reason why you couldn't do that for, more species across the tree of life. But at some point, I think that would start to break down and like you probably wouldn't be able to get all the way back to aligning like a bacterial genome. But yeah, to some limited phylogenetic scale, I think you could totally do that and it'd be very interesting to look at. It's a great question.
Yeah, very cool. Yeah, not necessarily the whole x-axis that you had. Yeah.
Yeah.
But and then one other question I had was were the in the cartoon the non-coding regions were located next to the close to those genes in the cartoon? Was that also how the analysis went? It needed to be adjacent or no?
Great question. Yeah. So I think if I'm remembering the number correctly, I think I defined that they could only be regulatory elements if they were within 2 kilobases of the start site of a gene. I think that's the cutoff that I used.
Okay.
Yeah. So they do have to be somewhat near a gene.
Okay. Yeah. Because I was just I was trying to brainstorm it because that's your definition of regulatory region here, and like what else is in the genome that could
Yeah.
be categorized that way that is also being included in that analysis. Do you know what else? I kind of can't think of it, but I would assume there's some other regions that are conserved that are also getting put in that category,
Oh,
which may or
yeah.
may not be regulatory regions. But I couldn't think of examples, basically.
Yeah, in terms of like what are other kinds of genomic elements that might be relevant to producing the phenotype, I can't
Yeah.
think of anything else. Certainly that definition of like it has to be within 2 kb of a gene
Hmm.
to be considered a regulatory element, that is a
Hmm.
somewhat arbitrary number. And like there are examples of genes that have regulatory regions that are like hundreds of
Hmm.
kilobases, span hundreds of kilobases upstream of the gene. And so that's kind of just an arbitrary cutoff. What I would love to be able to do in the future is make use of something like a TAC-seq and really get an
Hmm.
idea of where, real experimentally validated
Hmm.
regulatory elements are in these genomes. But yeah,
Gotcha.
maybe in the future.
Very cool.
Great question. Thanks.
A little more time if anybody has a last question. All right, it looks like we're good. Thank you,
Yeah,
Dr.
thanks so
Barkdull, and
much.
congratulations too.
Yeah, thanks
Again,
everyone for being here today. It's great to
yeah,
Yeah,
share my
thanks so
work.
much, Megan. Thanks so
ah,
much, Kelly, for hosting. And
yeah,
we'll see you guys next week.
at the next.
Awesome. Thanks so much.
Thanks,
Bye-bye.
Megan.
Bye.
Bye.